this invitation to what's been 2 interesting days of some amazing science and technology. One of the things that emerged in yesterday's discussion was at time the difficulty of taking the science to the end users, so let's say into society, and to actually being used. And that difficulty from the discussions emerged, well, there's lots of difficulties, but one of the main difficulties was funding. Investors are scared, and they're scared for their return. And what we've been doing at ICGB for nearly a decade and a half now is to try and take the fear factor out for any investor that wants to produce biosimilar drugs. So I'm going to speak a bit later about, or in more detail, what a biosimilar drug is, but basically it's just a biological, and you've heard about this yesterday, I think, in the morning talk. So a biological, so it's produced and extracted from a living organism, so it's distinct from normal pharmaceuticals that are chemically synthesized. The process is a bit more difficult. These types of drugs are a lot more complex. You can see in the picture here at the end, basically a comparison of the structure of a biological. This is a monoclonal antibody to, let's say, a normal chemically synthesized drug. We're obviously using 2 extremes. So here I'm using monoclonal antibody, which is one of the bigger biologicals, and aspirin. Another major difference really is in the price. Biologicals, due to the mode of production, due to their complexity in the purification from the living organisms, are really, really expensive. But they also have tremendous health benefits. And we can talk about interferons or monoclonal antibodies in the treatment of cancer, or homeostatic growth factors, just erythropoietin and complement stimulating factors. And it's quite lucky that I've decided to choose to talk about insulin, considering the previous talk. So insulin, before its recombinant form, was produced by extraction from pig parts. It used to take around 2 tons of pig pancreases to produce around 60,000 doses. And today, we can produce an equivalent amount of doses with a simple 250-litre fermentation of E. coli. And that's something extremely important, considering that today, there's around 350 million people worldwide suffering from diabetes. So if we won't be able to produce the recombinant form of insulin, it would be extremely difficult to treat the ever-increasing number of patients with diabetes. Now, notwithstanding, this was the first biological drug approved by FDA more than 35 years ago. It's still basically unaffordable to the majority of people on this earth. And this applies with all biological drugs. They're extremely expensive. Not everyone has access to them, and where you do have access to them, they do represent a significant burden on the healthcare system. And I think, and for example, remaining in the field of diabetes, this is a figure from the UK, the government spends around 1 billion on the diabetic drug per se. Another example of how they weigh on the healthcare system comes from Italy. So you can see that in Italy in 2014, I made a mistake in the figure there, we spent just under 4 billion euros on biologicals alone in the hospital setting, and that accounts for basically just under 40%, so it's 35% of total drug spending in the hospital, of all the pharmaceuticals the hospital are prescribing to the patient. So you have this bizarre situation where the minimum amount of pharmaceuticals given in the hospital setting are accounting for a much, much higher proportion of the cost. And that also exists within the biologicals itself. Now, this is an example of the volume and the cost of biologicals of that 4 billion. So you can see, for example, the therapeutic proteins such as erythropoietin, human growth hormone, GCSF, in terms of volume, account for around 90% of the drugs prescribed in the hospital setting. Monoclonal antibodies for the treatment of cancers or rheumatoid arthritis account for 6%. If we look at the cost, that 6% goes up to 45%. So again, this is an example of how access to these biologicals is quite difficult. And remaining in the monoclonal field, I'm going to use the example of anti-TNFs. Now, there's several anti-TNFs on the market. They are the blockbuster drugs. Sales were around 10 billion for Humira, for example. However, in this paper here, when they analyze how many people with rheumatoid arthritis are actually having access to the pharmaceutical, it's around 20%. No? So The reason for this is that a year's treatment costs around 60 to 50,000 euros. And so doctors aren't really allowed or are discouraged to prescribe the monoclonal and have to try alternative pathways first with small chemicals. What you really want to be doing, and this 20% is basically patients with severe rheumatoid arthritis that we treated. What you really want to be doing is to treat people with moderate rheumatoid arthritis to avoid them going to severe rheumatoid arthritis and still be able to function a normal life, work and function normally. So there's an occasion, a lot of these biologicals are now off patent or slowly becoming off patent, and they can be used as what I mentioned before as biosimilars. And this biosimilars really do provide the potential to reduce prices and expenditure. And below here, I've actually written what a biosimilar is. It's a definition of the European Medical Agency, so it's a biological medicine that's highly similar to the reference medicine, notwithstanding natural variability, which is something that all biological medicines have. Even the protein in our body also has the natural variability from one person to the other. And the other thing says there's no clinical meaningful differences between the biosimilar and the reference medicine. What do these definitions mean? It means basically if I can demonstrate, so on here you have like a normal pathway for clinical approval. So you have some analytical work and huge clinical studies. This definition here means if I can demonstrate analytically in non-clinical studies similarity to the originator product that is now off patent, I can probably reduce my clinical trials or have them extremely focused. And this is the major area of cost in taking a drug to market. Okay. This also creates this fact that it's called a biosimilar and cannot be identical to the originator creates some fear. It's taken a while for biosimilars actually to be accepted both by clinicians and patients. The variability though, why we call it a biosimilar, as I already mentioned, is due to the fact that we produce it in a living organism. So a living organism, the same strain grown here in Bratislava and the same strain grown in Trieste in our labs, will probably have some natural variations because we can't duplicate the process exactly. And lots of times they always show these zebras, so how similar is similar, they're all zebras but they're different. And what I'm trying to say is that variability is allowed. You don't have to be fixed on the fact that it has to be identical. It is a biosimilar, the European Commission, the Medical Agencies accepts it as a biosimilar, so all you have to do is try and make it as similar as possible to the originator. Even the originators vary over time. This is a publication that shows some chromatographs of pre, which is in blue, and post-production change. So the company had to expand its production, and you can see their product on the market changes between pre and post-production. So again, I'm just saying this to try and make people understand that variability is allowed. And as people are normally scared of biosimilars, I always show this one as well. Biosimilars in Europe. Europe's been the leading area in the world with regulation of biosimilars from 2005. So in 11 years, this number's probably gone up. Now there's been 28 approvals and 0 safety concerns. Okay. The possibility, well, these biosimilars have fulfilled their promise. On the top table, you can see price reduction in Europe. So, for example, erythropoietin, the biosimilar is around 30% reduced. Less price reduction with anti-TNFs. In India, the price reduction is even greater. That's the table on the bottom. The price reduction is around 60% for these monoclonal antibodies. So, the biosimilars entry into the market has eroded the prices and has reduced costs. And these costs also result in a better economic perspective. So you can see this is basically a graph of use of GCSF in the UK. So this is the originator, the line in blue. This is a modifying form of GCSF. It's pegylated, and this green line here is the biosimilar. So you can see whereas these two lines basically remain constant from 2008, I think it goes to 2014, the entry of the biosimilar has slowly increased. So we arrived to a point where nearly twice as many patients are treated with GCSF now than previously when the biosimilar wasn't on the market. And something really nice is that the increased treatment has been associated with reduced admission rates because obviously the patients are now getting GCSF, the white blood cell count is coming back to normal, so secondary infections are less. we've been doing at ICGB these last years is to try and increase or accelerate the availability of these products at more affordable prices by supporting local biopharmaceutical manufacturing in our member countries. And we do this by providing, let's say, a hub, a facility that provides high-yield cell lines, know-how in up and downstream processing, know-how in analytical methods and the quality control, reference reagent, and more important than anything, training at ICGB. So I'm not going to go into too much detail because you'll be hearing about this more tomorrow. So basically what we concentrated on, this is the triangle I showed you before, is this area here, the physical, chemical characterization, the biological characterization. So we're taking the fear, let's say, away from the investors by performing these process development, process analytics, back and forth, until we can comply with the quality necessary for the investor to feel comfortable to then take our technologies, go to their country, do the manufacturing, and then apply for regulatory permission to go to the market. I'm going to skip this one because this one's just a series of tests we do for the quality control. You'll be hearing more about that tomorrow as well. There'll be more practical examples of what we do in our labs. Well, I'm just going to say that this procedure is economical viable. So basically, this is a table that shows, let's say, a portfolio of the technologies developed at ICGB. I'm not going to go into all of them. I'm just going to use GCSF, for example. The process we developed produces around 10,000 doses. But what's more interesting for me is that the cost of production of this, in terms just of reagents, I'm not calculating building or wages, whatever, is, I can't read it, but I imagine it's 0.12 compared to the price of the drug, which is 136 euros in Italy. So basically, the cost of production in terms of reagent is less than 0.1% of the actual cost of the drug in terms of sales. So there's ample margin for production and entry of the market and competing with the other people that are already on the market. So this, let's say, idea or initiative that we had at ICGB several years ago has been extremely successful. Since 2001, we've signed 56 collaboration agreements with 22 different countries, with 22 different companies from 20 out of the 64 of our member states. You can see the countries in the world map below where we've transferred our technology. The ones in yellow are countries that have received the technology but still not on the market. We do have success stories. Those are the ones in green. These countries have taken our technologies and are presently on the market with their products. Equally important for us is that it's also allowed us to train scientists from our member countries in the procedures for pharmaceutical production and quality control of biosimilars. And I've been probably too fast and extremely brief, but what I want to say, and this is like the conclusion, is that competition in biosimilars, I hope I've shown you, or in biologics, through biosimilars, lowers prices of the pharmaceutical. It translates to increased accessibility. It translates into better health. And the more important thing is any investment by private, governmental institutions in this area increases the technology of the country, increases the scientific know-how. It gives you independence because most countries don't like to be dependent on the big pharma for their drugs. No? Investment will also decrease your healthcare bill for the service. No? It's going to be reducing the prices. They're going to be spending less on the pharmaceuticals for its population, and subsequently it will increase health of the population. So that is just really a very quick overview of why I think investment in biosimilars is extremely attractive. Tomorrow you'll be hearing more details about it from Natasha Skoko, and this is the team at ICGB that's dedicated to this initiative. Thank you.